Hi everyone, it's Dr. LeBlanc with another video screen capture lecture on RNA processing. The learning goals for this lecture are to understand that RNA transcripts must be modified before they are functional, to understand the modifications made to ribosomal RNA and to transfer RNA, to understand the two different mechanisms of self-splicing, to understand the mechanism of messenger RNA splicing by the spliceosome and the RNA elements in the messenger RNA. To understand end processing of messenger RNA in eukaryotes, both capping and cleavage and polyadenylation. And to understand the role of the C-terminal domain of RNA polymerase II in coordinating messenger RNA processing events. There are many different RNA modifications that eukaryotes undergo. Splicing, capping, polyadenylation, editing, and base modification. We're going to focus on splicing, capping, and polyadenylation, and not so much on these other two in this lecture. tRNAs, ribosomal RNAs, and messenger RNAs are all modified. Ribosomal RNAs have spacer regions removed to produce 18S, 5.8S, and 28S ribosomal RNAs from the same transcript. tRNAs are processed by exonucleases at the ends of a strand and endonucleases within a strand. Messenger RNAs are capped, spliced, and polyadenylated, and also sometimes they're edited. Okay, let's look at the ribosomal RNA transcripts first. Here is a diagram of a ribosome. A ribosome has a large subunit and a small subunit, and inside these large and small subunits are RNAs, transcripts. A 28S ribosomal RNA in the large subunit and a 18S ribosomal RNA in the smaller subunit. This S is a measure of density and shape, and it also denotes size. So 28S RNA is larger than an 18S RNA. In the DNA, there is a gene that encodes all three of these RNAs in the same gene. RNA polymerase I binds to the promoter in the DNA, and it transcribes one long piece of RNA that contains the sequences for all three of these RNAs. So here's the long transcript. Okay, it's a very long transcript. It contains the sequences for the 18S, the 5.8S, and the 28S RNA all within this transcript. And in between are spacer RNAs that are not functional. The light pink areas are cleaved to release an 18S ribosomal RNA, a 5.8S ribosomal RNA, and a 28S RNA. This process occurs in the nucleolus of the cell, a small area inside the nucleus that takes care of processing ribosomal RNAs. These are the functional RNAs that are key in ribosomes, including catalysis. The 28S RNA has catalytic properties that help produce the amino acid chains that are proteins, that fold up into proteins. One of the reasons, a good reason, to have the 18S, 5.8S, and 28S all part of the same transcript is that all three of these RNAs, which are necessary for one complete ribosome, are always produced in stoichiometric amounts. Having all three of these transcripts produced by the same, within the same initial transcript ensures that you never have more 18S RNA than you need or too little 5.8S RNA They're all produced in equimolar amounts. tRNAs are produced by RNA polymerase III. They are produced as a much longer transcript than their final functional product. So a large precursor is produced, which is then subsequently cleaved by an endonuclease, an RNase P. It cuts it right here and releases this portion of the RNA. And also another endonuclease releases this portion. An exonuclease will then trim away until you have this CCA end, which is the same end on all tRNAs, and this end is where the amino acid will get attached. In addition to trimming the ends, there are also bases that are modified. tRNAs are known to have many different types of modified bases, and these add stability, and also they add to the thermodynamic properties of the molecule to get it to fold up into the right shape. These are some of the different modified bases that you can find in tRNAs: methylguanosine, pseudouridine, 4-thiouridine, and isopentyl adenosine. Eukaryotic messenger RNA processing is complex. Before it leaves the nucleus, messenger RNA is spliced, introns are removed, it is capped at the 5' end, and a long stretch of adenosine residues are added to the 3'. While the messenger RNAs are still in the nucleus, introns have to be removed. Introns are non-coding regions in the middle of messenger RNA transcripts that separate exons from each other. There are 3 different types of ways to remove introns from messenger RNAs. 2 of them are self-splicing. Group 1 self-splicing introns and group 2 self-splicing introns are found in some lower eukaryotes and occasionally some mitochondria and some chloroplasts. This was one of the very first demonstrations that RNA had catalytic properties. In group 1 introns, the intron itself folds up into a 3-dimensional structure, grabs onto a guanosine nucleotide, and the 3' hydroxyl of the guanosine nucleoside attacks the 5' splice site, releasing a 3' hydroxyl end, which then in turn attacks the 3' splice site and releases the RNA intron. And the 2 exons of the messenger RNA are now contiguous and ready to encode a protein when it binds to a ribosome out in the cytoplasm. Group 2 introns, self-splicing introns, have a mechanism that's very similar to the spliceosome, which we're going to see in the next slide. Group 2 introns use an internal adenosine residue, and the 2' carbon with that reactive hydroxyl group on it attacks the 5' splice site to form a circular structure within the intron. This free 3' hydroxyl now attacks the 3' splice site of the intron and releases this lariat structure that has a circular portion and a linear portion. And the 2 exons of the messenger RNA are now contiguous and can direct the synthesis of a protein when it binds to a ribosome out in the cytoplasm. Most commonly in the nucleus, messenger RNAs are spliced by an elaborate machinery called the spliceosome. The spliceosome has the same chemistry as the group 2 self-splicing introns, but it uses a lot of accessory proteins and RNAs to get the job done. So that an internal adenosine residue has a 2' hydroxyl, because it's an RNA molecule, that attacks the 5' splice site, makes the circular portion of the intron. This free 3' hydroxyl attacks the 3' splice site, and this lariat structure is released, and the messenger RNA is now ready to synthesize a protein. Let's look at some of these individual steps of splicing. So here we have a transcript, a primary transcript, just as it came off of the DNA template. There are exons and introns, or intervening sequences, that do not encode amino acids and that have to be precisely removed to preserve the codon structure of the messenger RNA. Every intron in eukaryotes has at its 5' site and at its 3' site the nucleotides GU and AG. GU-AG. These are nucleotides that are part of the introns right at the 5' splice site and the 3' splice site of the intron. In addition, they all have an adenosine residue at the branch site. What happens when an intron is going to be removed from a messenger RNA is that a small ribonucleoprotein particle called U1 binds to the 5' splice site, and another small ribonucleoprotein particle binds to the branch site right here. Okay, a ribonucleoprotein particle contains protein and small RNAs, and those small RNAs are critical in positioning these particles at the right spot. The U4, U6 complex and U5 trimer bind next, and the intron loops out, and the exons are brought closer together. These are all the different components where they're located. The 5' splice site is cut first, and the 5' end of the intron is connected to the A and the branch site to form a lariat. We saw the biochemistry of that in the previous slides. Some of the ribonucleoprotein particles are released, and then the 3' splice site is cut. Exon 1 is connected to exon 2, and the intron is released and subsequently degraded. This is a slide just showing the different occurrences of group 1, group 2, and pre-mRNA introns. Pre-mRNAs are the ones that we're most interested in. They're removed by the spliceosome, and they're commonly found in structural genes within the nucleus of eukaryotes. Almost every single eukaryotic gene has at least one intron. There are some rare exceptions to this. For example, the histone proteins of the nucleosome, their messenger RNAs do not have introns, but for the most part, every single eukaryotic messenger RNA has at least one intron. These group 2 introns are found in some tRNAs, ribosomal RNAs, some fungi and plants, and chloroplasts, and some bacterial genes, but very rarely. And the group 1 introns also in some lower eukaryotes, in tRNAs, and some mitochondria, and chloroplasts as well. One of the big puzzles of the Human Genome Project was the fact that there were so few genes contained within the human genome. There were only about 20,000 to 25,000 genes were able to be identified in the 3 billion base pairs of DNA that make up a human genome. But the complexity of humans led to the conclusion that there must be some other way to produce all the proteins that are necessary to make up a human body. Where do they come from? How can you account for the fact that there are so many different proteins and so few genes? Well, one mechanism to generate protein diversity is to encode several different proteins within a same gene. And the mechanism that is achieved by is called an alternative splicing. Here is a very good example of how one single gene can produce many different proteins. Here is a gene for alpha tropomyosin. It is a protein that is found in muscle tissue. Here is the primary transcript of the gene. It contains several different exons, 1A, 2A, 2B, 1B, 3, 4, 5, 6A, 6B, 7, etc., all the way down to 9D, and many intervening sequences. The intervening sequences here are in light green, and the exons, the parts of the genes that encode amino acids, are the colored parts, the other colored parts. So this one gene can produce 1, 2, 3, 4, 5, 6, 7, 8, 9 different proteins by mixing and matching the exons contained within the gene. In order to get certain of these exons linked together to produce a different polypeptide chain, what has to happen is the introns have to be removed in a specific order and a specific pattern. This is called alternative splicing. So, for example, the alpha tropomyosin that's found in striated muscle contains exon 1A, exon 2B, exons 3, 4, and 5, 6B, 7, 8, 9A, and 9B to produce a polypeptide that's unique and not found in other tissue. Alternative splicing has skipped over this exon and this exon and this exon and not included these end exons. A very different protein is found in the brain. Instead, it has exon 1B, 3, 4, and 5, 6B, 7, and 8, skips the 9A and 9B and just has 9C and D at the end. So we have two very different versions of alpha tropomyosin in striated muscle and brain tissue produced from the same gene by skipping over exons or alternatively splicing the exons together. The 5' ends of messenger RNAs and eukaryotes are capped to protect them from degradation as they make their transit from the nucleus into the cytoplasm. This is the process by which the cap is added. This is the first nucleotide of the messenger RNA. It has a 5' end and a triphosphate. This is it right here. Okay. What happens first is a triphosphatase removes one of the phosphates, and then a guanylyl transferase hydrolyzes the GTP. And the GMP is attached to the 5' end, and pyrophosphate is released. Okay, so we added some more phosphate groups here. Here's a new nucleotide being added. And finally, a little methyl group is added onto this 7-methyl at the 7 position. to create the 7-methylguanosine cap. So here's the guanosine right here, attached by a triphosphate to the first nucleotide of the messenger RNA. This fancy cap structure protects the messenger RNA from being degraded by exonucleases. At the very end of the transcript, we have a polyadenylation signal sequence, AAU, AAA. This is found in all Pol2 transcripts. An endonuclease comes along that cleaves in the middle of the sequence of RNA that's at towards the 3' end, about 20 nucleotides downstream from the AAU, AAA, and then polyadenylation occurs. An enzyme called poly-A polymerase adds an A and an A and an A, up to 200 different A's are added at the end of the 3' end of the messenger RNAs, and that also protects it from degradation. In addition, there's poly-A binding protein that binds to the poly-A tail to further protect the end from degradation. So we have capping, we have splicing, we have polyadenylation, 3 major modifications of the messenger RNA transcript that occurs in the nuclei of eukaryotic cells. This does not occur to RNA transcripts after they are produced, but it occurs during the process of RNA transcription. Here's RNA polymerase, here's a DNA molecule, here's the start site of transcription. So here's a gene right here, and here's RNA polymerase producing a messenger RNA. This long squiggly purple line is the C-terminal domain of RNA polymerase. Notice that it has phosphates attached to it. When the 5' end is first produced, the CTT has one pattern of phosphorylation, and the capping enzymes bind. Later on, the pattern is changed, and the RNA splicing machinery binds, and later on, the pattern of phosphates is changed again, and the poly-A tail machinery binds. We're going to look at each one of these individually. So, here's RNA polymerase. The 5' end of the messenger RNA is just produced, and the capping enzymes bind to this C-terminal domain and add the 5' cap. You can think of this as kind of a platform that the different machinery is going to load on, do its work, and then load off again. Each time you switch the different types of machinery, the phosphorylation pattern of the C-terminal domain is changed. So here we have a different phosphorylation pattern now. We've already capped the end of the messenger RNA, and now we need to splice out these introns. So the splicing machinery loads onto this platform and does its job. And finally, we switch the phosphorylation pattern of the CTD, and the poly-A tail machinery comes on, and it's going to cleave the RNA and add the poly-A tail onto it. Here are some other videos you might like to watch to get another perspective on some of these processes. So, in summary, RNA transcripts must be modified before they are functional. Ribosomal RNAs are produced as one long transcript containing the 28, 18, and 5.8S ribosomal RNAs, and then they're cleaved by endonuclease. This ensures the production in stoichiometric amounts of all these RNAs, one ribosome set per transcript. Long transcripts containing tRNAs are cleaved to produce the final 5' and 3' ends, and they also undergo extensive nucleotide modification. Group 1 and Group 2 self-splicing introns are found in prokaryotes and involve the removing the intron and the linking of the exon by a phosphodiester bond. It was one of the first examples showing that RNA had catalytic properties. Spliceosomes remove messenger RNA introns in a similar biochemical mechanism to the group 2 introns. The spliceosome is made up of several small nuclear ribonucleoprotein particles containing both protein and the small uRNAs. The invariant consensus sequence, GUAG, are found at the beginning and the end of every eukaryotic intron. GU at the 5' end of the intron and AG at the 3' end. And in between is an A. The adenosine residue in the middle of the intron forms a branch point in the first step of the splicing reaction, and the intron forms a lariat structure, a loop with a tail. As the intron is removed, the exons are joined by a phosphodiester linkage. The sequence of nucleotides maintains the open reading frame, which we're going to talk about when we talk about protein translation. The 5' end of eukaryotic messenger RNA is modified by a 7-methylguanosine cap and the 3' end by a poly-A tail. Both of these modifications lend stability to the messenger RNA as it travels from the nucleus to the cytoplasm, where it is translated into protein. All of these mRNA modifications take place sequentially on the C-terminal domain of RNA polymerase II. The C-terminal domain of RNA polymerase II undergoes rounds of different phosphorylation patterns, resulting in the different processing machineries to be loaded onto this platform sequentially, first capping, then splicing, and finally polyadenylation. This is a very good table showing the differences between transcription and RNA modification in bacteria and eukaryotes.